Welcome to our talk on the molecular smart switch that unlocks protein function using the auxin-inducible Degradation system, illustrated here by a simple on divided by off switch entwined with a protein. Over the next slides, we'll explore how this tool gives precise, rapid control over protein levels and why that matters for understanding cellular biology. Proteins control nearly every cellular process, but their function changes over time and space, as the diagram shows, linking cell cycle, signaling, and developmental patterning. To truly understand a protein, we must observe what happens when it's removed at a specific moment and place, not just permanently, because timing reveals primary roles versus downstream effects. For decades, gene knockouts and RNA interference were the main ways to probe function, but they can be blunt instruments. Knockouts are often irreversible and lethal for essential genes, while RNAi can be slow, incomplete, and produce secondary or off-target effects, making it hard to tell direct roles from adaptive cellular responses. Instead of targeting DNA or mRNA, targeted protein degradation cuts straight to the protein itself, as the graphic contrasts indirect and direct control. This approach delivers faster, more precise perturbations, letting us observe immediate consequences of loss of function and thereby distinguish direct protein roles from long-term cellular adaptations. The auxin-inducible Degradation, or AID system, borrows a plant hormone pathway and requires two pieces: a small Degradation tag on the protein of interest and the plant F-box protein tier 1. In the absence of auxin, the protein is functional, but adding auxin rapidly recruits ubiquitin ligases and triggers swift, reversible degradation, giving tight temporal control. A classic case study asks whether GRASP55's role in Golgi structure is direct or secondary. Slow RNAi produced ambiguous defects, but the AID approach depleted GRASP55 in under 2 hours, and no immediate Golgi stacking defects were observed, revealing that previous RNAi phenotypes were likely secondary consequences of prolonged depletion rather than primary functions. RIF1 illustrates how timing matters during DNA replication stress. Older methods couldn't separate roles during versus after stress, but AID allows depletion either while stress is ongoing or only during recovery. Experiments showed RIF1 is essential in both phases, revealing temporally distinct functions that were previously inaccessible with slower perturbations. Studying the ubiquitous dynein subunit, DHC1 in C. elegans highlights tissue-specific power. A global knockout is lethal, but by expressing tier 1 only in certain tissues, we could deplete DHC1 locally and uncover distinct roles in the pharynx, gut, and germline, demonstrating how AID enables in vivo tissue-level dissection of essential proteins. Condensing complexes shape mitotic chromosomes in a stage-specific way that was obscured by developmental lethality in traditional knockouts. Using AID to rapidly remove condensin at a defined cell cycle stage revealed a hierarchical loop formation model, where condensin II builds large outer loops, later subdivided into smaller loops by condensin I during mitosis. CTCF's influence on genome folding can be tuned rather than binary, and the AID system makes that possible. By varying auxin concentration to partially deplete CTCF, researchers observed dose-dependent changes in chromatin, insulation, and folding, turning a simple on-divided by off model into a quantitative rheostat for probing biological relationships. The original AID had two major drawbacks: leaky degradation without auxin, and the need for very high auxin doses. AID 2 addresses these issues with engineered TIR1 divided by auxin pairs and alternative F-box proteins, dramatically reducing background loss and lowering auxin requirements, which enhances specificity and minimizes off-target cellular effects. When choosing a degradation tool, trade-offs matter. DTAG and Halo Protex offer simplicity or different kinetics, but the AID system stands out for its broad model organism support and unique ability to be made tissue-specific in vivo. That versatility makes AID particularly powerful for developmental and organism-level studies where spatial control is critical. Targeted degradation goes beyond small molecule inhibition by eliminating the entire polypeptide and all of its functions, catalytic or scaffolding, as the schematics show. This universal approach reduces ambiguity from partial inhibition, and real examples, like studies in Plasmodium, have clarified true protein requirements that inhibitors had masked with secondary effects. Looking ahead, the future of functional genomics is on-demand and dynamic. The AID system delivers speed, specificity, temporal precision and tunability, and combining complementary degradation platforms could allow simultaneous, orthogonal control of interacting proteins, moving us toward a complete, controllable map of proteome function in living systems.